Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is now lecture 51, where I will talk about few interesting case studies on manipulating the metabolic pathway of lignin. So, that is why metabolic engineering of monolignol pathways. So, in the previous class I have discussed in detail about the classical and contemporary views of monolignol biosynthesis. I have mentioned the role of different enzymes which basically makes this monolignol formation possible. Also, I have discussed about the possibility of new lignin components which may come from caffeoyl moiety as well as 5-hydroxy moiety, and so I hope it is clear to you. So, now with this I go to the case studies. So, an outline of monolignol biosynthesis I am not discussing here. At the end I will again bring the slide. So, let us specifically go to the specific case studies. So, this one is basically manipulation of lignin quality by down regulation of cinnamyl alcohol dehydrogenase. So, you see that cinnamyl alcohol dehydrogenase where CAD will function. So, CAD is basically converts the coniferyl aldehyde or cinnamyl aldehyde into coniferyl alcohol and or cinnamyl alcohol. So, this is the enzyme which is called CAD, which is called cinnamyl aldehyde dehydrogenase. So, this is a generic name. So, that makes both the reaction. So, CAD, and I have recently mentioned that now there is an evidence of SAD, and if the lignin is dominated by this eventually makes the guaiacyl, GUAIACYL, and this makes the syringyl component. So, it is a G and this is the S. So, S is to G ratio is important for the lignin quality and also for the delignification issues. Okay. So, what they have done here, that they have down regulated the plant, model plant was used tobacco, and they have down regulated the gene by using this antisense expression of this CAD. So, so manipulation of lignin quality by down regulation of cinnamyl alcohol dehydrogenase. So, what is going to happen, that when the CAD activity is blocked or suppressed, then this leads to less accumulation of coniferyl alcohol or cinnamyl alcohol as appropriate, or it is also possible because if you think about the new views. So, new views, what I have said, although we will explain this from the new views, although the paper published in 1998, that time it was not clear at all. So, what we know, that from coniferyl aldehyde cinnamyl aldehyde is formed, right. So, it is possible that by blocking CAD here, the pathway may flow in this direction, okay, leading to the formation of more of syringyl lignins. It is possible. Okay. So, what is going to happen, that although the lignin content will be reduced, that is the most important point, because you are blocking the CAD, but it will affect the lignin quality. That is why this terminology was used, and what has happened, that these two are the two antisense line. So, antisense line 50 is more stronger. So, more suppression was worked, and if you see, this is basically the control. Okay, and if you compare this with this, what you see here, clear changes in the coloration of the lignified portion. Okay. So, this was further studied with different staining, and what has been found here, that this looks more intense as compared to other, and also here. So, and when they check for the CAD activity, you see in the antisense line the CAD activity is minimum when they compared with the control. So, that means indeed CAD suppression occurred, and as a result of that there happened to be, or there happened changes in the lignin quality, which is reflected from the cross section viewed under microscope with different staining. So, and in this paper what they said, basically this is the way the lignin quality can be manipulated, and particularly you can think ahead about the wood quality. So, the wood coloration, this discovery can really make contribution to the wood coloration, or novel wood colors can also be created by transferring this gene in an appropriate pulp paper producing plant such as aspen or so. The next one which I am going to discuss, this is another very interesting paper which published in Nature Biotechnology in 1999. So, the title says that repression of lignin biosynthesis promotes cellulose accumulation and growth in transgenic trees. So, what they have done, as I have shown you, that very briefly I will tell you the pathway. So, cinnamic acid makes paracumaric acid, and paracumaric acid converted into paracoumaroyl CoA, and then paracoumaroyl CoA either it makes coumaryl aldehyde, or it may makes caffeoyl CoA, and eventually that contributes to G residue, S residue, and this is H residue. Okay. The enzyme here is 4-Cl. So, what they have done, here they made antisense suppression of this 4-Cl. So, the plant was used is the target and plant or gene source is same, the poplar, or no, is aspen is the English name, which is called Populus tremuloides. This is a plant which is used for making pulp and paper. So, they created transgenic aspen with antisense expression of PT 4-Cl. PT stands for Popular tremuloides. So, what they thought, this may be the best step to block the pathway. Okay. Now, one thing I mentioned, that the interconnections between the pathway, that is meta hydroxylation, converting caffeoyl, converting coumaryl to caffeoyl. So, this was discovered in 2004, 5, 6 onwards. This paper was published in 1999. Even then, these scientists from Michigan State University, led by Vincent Chiang, who was the leader in discovering these pathways. So, they thought such a nice possibility that blocking 4Cl at the beginning will make impact into the end product. Now, question which came to our mind that there are multiple 4Cls. Now, in fact, the 4Cl what they have chosen, that is specific for paracoumaryl CoA. So, this 4Cl is actually 4-hydroxy cinnamyl CoA ligase, but specific for paracoumaric acid. So, that it accepts paracoumaric acid as a substrate and make paracoumaryl CoA, not the ferulic acid as a substrate or synapic acid as a substrate, that you must remember. So, this is the beauty. And then what we see, that what has happened as a result of these, what has happened, the outcome significant reduction in lignin contents. Let us see. So, this is the pathway. So, this is the 4Cl here. These 4Cl they have blocked using antisense PT 4Cl. So, when this is blocked, then what will happen? The flow, and I have mentioned by now it is clear to you that this pathway as such is not contributing towards monolignals. Okay. I have mentioned that. So, what is going to happen, that here basically the flow when you block here will be reduced, and if it is reduced, then definitely the one flow which goes to H, that will be also reduced. The flow which goes to G, that will be also reduced, and the flow which goes to S, that will also reduce. So, as a result of that, what they have found, repression of lignin biosynthesis. But interesting point is that this promotes cellulose accumulation and the growth of the transgenic trees. So, I will show you some of their results. So, here this is the control plant. So, where no normal 4Cl is functioning, no expression. So, these 4 are different transgenic aspen expressing antisense 4Cl. What you see here, the growth you see is much more in all the transgenic aspen with antisense expression. That is surprising, but that is what happened. So, it did not really affect the growth, but it reduced the lignin content, but compensated with cellulose accumulation, and that is why that is reflected in the growth. Now, as I said, the repression of lignin biosynthesis, that is beautifully shown here in this left side. So, what they have done, basically fluorescence microscope of the cross sections of a nodal portion of the transgenic and the control line. So, this is basically the control, and this is basically the antisense expressed transgenic line. So, they have used agrobacterium tumefaciens mediated transformation, but I am not discussing that, because that is not within the purview of this course, and I have mentioned about how transformations are being done. So, we are interested only to see the consequence of genetic manipulation. So, here you see in the control with this ultraviolet excitation at 365 nanometer, it shows autofluorescence. So, autofluorescence is the characteristics of the phenolic compounds. So, the autofluorescence you see here xylem, but here the autofluorescence level is low. Why it is so? Because it suppressed the lignin formation. This is very clear. And the next point is that it promotes cellulose accumulation. So, you see that this is how this is a diagram I put it here. where lignin or cellulose actually they form the complex in the cell wall. Okay. So, what happens that when cellulose and hemicellulose are also there. So, when lignins are suppressed, that somehow compensated by cellulose enhancement. How it is happened they are unable to explain, but it happened, and as a result of that, whether it is good or bad, it is indeed good, because more cellulose accumulation as a result of compensatory mechanism. It is good because that makes the plant aspen, transgenic aspen plant more suitable for pulp and paper making. Why? You know, the paper quality is dominated or determined by the cellulose quality. If the cellulose content is more, the paper quality will be more. And also another important point is that in order to make the pulp and paper, one need to delignify. So, delignify means the wood has to be treated with harsh corrosive chemicals, which are not environmentally friendly like caustic soda and so. So, that will delignify the wood. So, now once the lignin content is less, so the treatment, delignification treatment will also be reduced. So, that will be environmental friendly, and second point is that it is producing more cellulose. So, actually it is basically a gift of the scientific venture. So, which can be indeed used for making pulp and paper, and that is why this paper published in Nature Biotechnology. So, Nature Biotechnology journal, outstanding papers with possibility of practical utility, such papers are published, and this is indeed outstanding, although it is pretty old, but in a class, even in my theoretical, in my classes where I do the board work, I always bring this paper instead of discussing some of the latest papers, because it is not the year of discovery important or the latest paper, it is important the scientific content. So, this is what it is. So, and the other aspect, what is this? This the same group. This is the group basically led at Michigan State University by Vincent Chiang. So, their group also published another paper in 2003 in Proceedings of National Academy of Science PNAS, where they have done three things. Number one, PT4Cl antisense suppression. Number two is this CALD5H overexpression, and number three, simultaneous expression of both PT4Cl antisense plus PT CALD5H sense. So, PT4Cl, if you again use a strong promoter, the level of lignin reduction will be more. So, that led to the formation of desired results, that is a significant reduction in lignin contents. And PTCLD over 5 overexpression, what happens, that basically leads to this one, this leads to more accumulation of, actually this is the online class, had it been a offline class, then I could have asked the same questions to the student, and students seeing the board they can guess and give the answer, but here you whatever I tell you have to listen. So, more accumulation of S units, is not it, because this contributes to the syringeal unit and this contributes to the G units. Now, when you make over expression, so that means the pathway, I have made a wrong marking. So, it is not this one, it should be the this one. So, that means the pathway moves this direction and eventually makes more S. So, more S has some advantage, because that will be easier to delignify if the S component is more than the G. So, that is the intention. So, that has been achieved. And the third one what they have done, so there they block this 4Cl. So, and then they over express this one, and as a result what is going to happen I will try to tell you. So, the result is this, that plants expressing both transgenes resulted in 52% less lignin and 64% higher SG ratio. S is first. So, the S is dominated and 30% more cellulose. It is a very ideal situation. So, that is what happened. And SG ratio increase also accelerated cell maturation in stem secondary xylem, pointing to a role of syringeal lignin moiety in coordinating secondary wall biosynthesis. So, what happens when this happens, that is more SG ratio, that actually leads to accelerated maturation, that is wall thickening and lignification, and that happened because of this CLD5 upregulation. And what you see here, this is basically the control and this is basically the transgenic. So, here what they said, that this leading to accelerated maturity here than compared to this one. And also what you see, that this is basically the control the lignin content was more, but in the transform lines where this 4Cl was suppressed, you see that the lignin the 4Cl activity is getting suppressed, and not only that, the lignin content which is represented here, the lignin contents the lignin contents are also getting suppressed, and lignin is to SG ratio is also getting enhanced, and the top one, the middle one is basically the enzyme activities. Enzyme activity for when it is suppression, then the activity of 4Cl is less as denoted by the red bars, and the activity of the CLD5H enhanced as a result of overexpression. So, what has happened, that this basically makes accelerated maturation with more of S component, that is useful for the industrial exploitation of these plants for making pulp and paper with less effort for making delignification and with a more S component, and that is also more of cellulose accumulation. So, that is all good. So, this is the paper which published in PNAS. So, here they have used 3 different ways, either singly or both together. So, the last one now I will discuss, that is the case study where down regulation of hydroxycinamyl CoA secumate hydroxycinamyl transferase. This is what is the HCT. So, the HCT down regulation, and that affect lignification, development and forage quality. So, HCT what it does? It basically converts coumaroyl CoA to caffeoyl coumaroyl secumate. So, coumaroyl CoA to coumaroyl secumate. Okay, let me use in that way. And coumaroyl CoA converted into caffeoyl secumate, and then it is converted into caffeoyl CoA. So, what is clear here? So, down regulation of HCT in the transgenic alpha alpha affects this. So, what they have done is basically they have used the antisense suppression or antisense expression of HCT, and what will happen if that is reduced, then this flow will be affected. So, this flow, if this flow is affected, that means coumaroyl CoA will accumulate. Okay, but the flow from this towards this will be less, and eventually the other lignin pathway will be less. So, then what is going to happen? One thing is that if you make antisense suppression of HCT, it is very important, because that will leads to what? Okay, space is less, I put it here, that will leads to less G residue, even less S residue, because you are controlling at this point HCT, because HCT is the point where which forms coumaroyl secumate from coumaroyl CoA. Coumaroyl secumate converted into caffeoyl secumate by C3H, that again converted into caffeoyl CoA, then caffeoyl CoA converted into ferulyl CoA, then ferulyl CoA subsequently utilized for pushing the pathway towards either coniferyl alcohol or via coniferyl aldehyde to coniferyl aldehyde 5 hydroxylase to eventually the synapyl alcohol. So, all route goes through this, and if all route goes through this, and if you make changes here, then obviously this will be affected. So, the lignin content is significantly suppressed, that is the outcome. But one more thing you have to understand, that you are allowing in this way to accumulate more of paracoumaroyl CoA. So, paracoumaroyl CoA level will enhanced. So, if paracoumaroyl CoA level will enhance, there may be a possibility that the pathway may move, that is which contributes for the formation of what? Which contributes for the formation of coumaraldehyde and eventually para coumaryl alcohol. So, once is accumulated, so it is possible that this pathway flow moves in this direction and this direction. Eventually, what will happen, that para coumaroyl alcohol will accumulate more, and that will contribute towards the formation of H lignin in higher amount, whereas this will be lower amount. So, the plant which was used, which is legume trifolium. So, that plant, okay. So, alfalfa, yeah, alfalfa trifolium is the scientific name. So, the alfalfa plant is basically, it is a dicot, it is supposed to produce more of S and G, but now because of antisense suppression it suppressed the total lignin content, but the composition got changed. So, it is now accumulating more of H lignin. So, when more of H lignin is accumulated, whether it is good or bad, that is the question we need to understand, because forage quality. So, this is used as a forage crop. Forage crop means cattle feed. So, normally cattle and sheep they prefer to eat grass, but they less prefer to eat dicots. Okay. Whereas, the alfalfa is used as a forage crop. Now, what happens that if the problem is this, that if the forage crop contains more of S and G residue, that is the lignified portion, that actually eventually helps, that eventually makes difficulty by this grazing animals to digest. As a result of that, they are facing indigestibility, leading to a disease called blood disease, and that eventually kills the animal itself because of more lignin content in their feed. So, that is why there is an effort to make less lignified forage crops, so that the cattle or grazing animals can safely eat this. Now, this is something very interesting here. What happened in case of this alfalfa, now the G and S component are reduced because you are blocking at HCT, but that in turn leads to more coumaryl CoA accumulation, and that coumaryl CoA channelized towards this H lignin pathway. So, where you have this CCR and CAD, and eventually it makes paracoumaryl alcohol, and it makes more amount of paracoumaryl alcohol, and that is now channelized towards the H lignin. So, eventually the transgenic alfalfa accumulating H lignin in higher amount, that is actually very good, and people have checked, the scientists they checked that in vitro digestibility, they found a positive results. So, it is basically promoted as a very appropriate forage crops with less lignification through this transgenic approach. And this paper published in Phytochemistry in the year of 2007. Again, I must say there are more latest papers, but I prefer to discuss this because its scientific implication is much higher than only the technological papers. And this is finally the picture. So, CK stands for the control, these are all control, and these are the transgenic. Although what happened that the plants appear to be suppressed, it is not like the aspen. Aspen, what happens that the transgenic plants are more healthy, but here no, they are bit less healthy, less suppressed. But the good point is that whatever they are, they the presence of this syringyl units, which is dominated, which is shown here by this purple red coloration, you see that that is now getting changed. So, that means it is not accumulating the S lignin. So, this is basically the transgenic, and this is the control. So, the relative levels of S were strongly reduced, although G units were reduced by similar. However, more striking compositional changes most striking composition is increase in the H unit, and I said the implication of that. So, with this, and this is basically the content of the lignin acetyl bromide digestion. These are the control, and these are the several antisense transgenic lines. It is suppressed, and monomer yield also they have shown. As you see here, that in the transgenic line, the yield of H is more than that of the control. Black one, which is more, whereas the others are suppressed. Both G and S are suppressed, but H is enhanced. So, it is a wonderful scientific achievement, and that is why I have covered. And with this, with three or four different case studies, I have given you some flavor of metabolic engineering of the lignin pathway. So, with this I end the class. So, basically before I end, just to tell you that this HCT was once was targeted, C4H was targeted, and the results were really outstanding, and these are the different units. So, this is basically the biosynthetic pathway what we have also studied, that the complexity, but complexity gives new tools for manipulation. With this I end this class. Thank you.